Good afternoon, everyone. Let's start with our technical session 3. Now I request Shreya Ravishankar Shukla to present her paper on estimation of antibacterial activity of curd against foodborne and human pathogens.
Hello everyone, my name is Shreya Shukla from ZSCT Thakur Shamnarayan Degree College, EYBSc Microbiology. I am working under the guidance of Mr. Kunal Thakur, Assistant Professor, and Coordinator Mr. Udayban Yadav. So my topic is antibacterial effects of curd against foodborne and human pathogens. So study was undertaken to access the antibacterial efficacy of lactobacillus acidated from curd and human milk samples. There are many species and strains of lactobacillus such as Casei, Fermentum, Plantarum, Pentosis, etc. Antibacterial activities of all the lactobacillus acidates were estimated by agar-well diffusion assay. Various human and foodborne pathogens such as Escherichia coli, Staphylococcus aureus, Salmonella typhi were checked by loading the curd samples in the Petri plate and checking the inhibitory effect against them. Curd is made by the process of curdling, which is the coagulation of milk. Curd consists of lactobacillus that produce lactic acid. These bacteria denature milk protein casein so they clump and coagulate to form curd. Lactic acid bacteria has 12 genera included in a group designated as lactic acid bacteria because of their ability to metabolize relatively large amounts of lactic acid from carbohydrates, such as Lactococcus, Leuconostoc, Pediococcus, Streptococcus, Lactobacillus, etc. So these are the images of Lactobacillus under electron microscope and under light microscope. Methods and materials. Medium used in this experiment were sterile agar plates, sterile McConkey's agar plates, sterile Salmonella shigella agar plates. Bacterial strains of bacterial isolates of curd were taken as subjects for the studies. Curd samples were collected from households of Mumbai, India, and also branded curd samples were taken. These three pathogenic bacteria strains of S. aureus, S. typhi, Escherichia coli were taken from the culture collection center. Isolation of bacteria was done and results were observed the next day. Antibacterial effects of properties of lactobacillus from curd isolates were done by using agar-well diffusion method. Well was formed by forming the Paul borer by using the Paul borer, and hence this method is also called Paul borer method. Plates were kept under isolation for 24 hours at 37 degrees Celsius, and the zone of inhibition was checked. Observation. So here are the observation of E. coli growth on McConkey agar, Aureus growth on nutrient agar, and S. typhi on SS agar. Results were that no zone of inhibition was observed on the plates for all the three samples, blood samples and pathogens. E. coli has normal microflora of human intestine, so it doesn't have effect on growth of E. coli. Conclusion: no zone of inhibition was found on each of the plates, hence it indicates that no antibacterial property was shown by the lactobacillus. These are the references for my paper. Thank you.
Now I request Shravani Rane to present her paper to estimate the effect of petrol on common bacteria.
Hello everyone, myself Shravani Rane. student of third year microbiology department of ZS City's Thakur Shamnarayan Degree College. My topic for research is effects of petrol on labs bacteria under the guidance of Mr. Kunal Thakur and our coordinator Mr. Udayban Yadav sir. So let's begin with an introduction. As we know, petrol is a combination of aliphatic and aromatic hydrocarbon that is a volatile liquid. In addition to frequently being used as an industrial solvent, a thinner, and fuel for internal combustion engines, it also has other uses. Next is adaptation of E. coli. E. coli is a facultative anaerobe, that is, it can degrade the hydrocarbons present in the petrol as a fuel. ATP is produced in E. coli through both oxidative phosphorylation and glycolysis when glucose is present as the carbon source. There are three stages. First one is isolation of biofertilizer producing microorganisms. Next is isolation of E. coli. And the last one is isolation and identification of E. coli. The materials and methods used for this are isolation and characterization of E. coli in degrading the hydrocarbons of petrol. Next, culturing of microorganisms, processing of carrier material, mixing the carrier and broth culture, packing, and the last one is proper storage. Observation. After the minimal inhibitory concentration, the dilution of the organism and the petrol was subjected to the viable count and the growth of E. coli was observed. It was observed that E. coli is capable of degrading the petrol. Here are the results of E. coli and organism collected from silencer. The minimal inhibitory concentration. Here comes the conclusion. Most of the previously isolated bacteria are known to grow at 30 degrees Celsius in the presence of lower petrol concentration, that is 0.005%, and exhibit delayed degradation. Within 21 days, the E. coli was found to degrade the petrol slowly and can be used as a source of degrading the pollutants. E. coli is capable and can withstand the petrol and can grow by the use for it. Thank you.
Now I request Lovely Mandal to present her paper, which is estimation of effect of smoke on normal human mouth microflora.
Greetings to all. I, Miss Lovelyn Mandal from SYBSCID from Thakur Shamnarayan Degree College, working under the guidance of Mr. Kunal Thakur, Assistant Professor of ZS City's Thakur Shamnarayan Degree College, going to represent my topic, Effect of Smoke on Normal Human Mouth Microflora. Cigarette smoking is a public health problem. It decreases the commensal population of normal microflora of oral cavity. The study was designed to determine the changes caused by tobacco smoking on the microbial population. Smokers were enrolled for the study. Oral swabs were collected from the oral cavity of the subjects. So, मेरे को वापस स्टार्ट करना पड़ेगा. हाँ ठीक है, मैं वापस बोल रही हूँ, बट ये जो है ना, ये थोड़ा ऊपर कर. मेरे को पढ़ने को नहीं हो रहा है. पढ़ने को नहीं हो रहा. कैसे करते हैं? क्या इतना ऊपर मत कर, नीचे कर. थोड़ा सा नीचे कर, थोड़ा सा. ठीक है. अबे ये क्यों नहीं हो रहा यार? अस्मी इधर आ बता तू आ। Greetings to one and all. I Greetings to one and all. Me, Miss Loveline Mandal from SYBSCID, ZD City's Thakur Shamnaran Degree College, working under the guidance of Mr. Kunal Thakur, Assistant Professor of ZD City's Thakur Shamnaran Degree College, going to represent the topic of effect of smoke on normal human mouth microflora. Cigarette smoking is a public health problem. It decreases the commensal population of normal flora in the oral cavity, leading to an increase of a pathogenic microbes. It causes oral cancer, color change on the teeth, and other health implications. The study was designed to determine the changes caused by tobacco smoking on the microbial profile and oral health condition of cigarette smokers. 120 subjects comprising 60 tobacco smokers and 60 non-smokers were enrolled for the study. Oral swabs were collected from the oral cavity of the subjects using sterile swab sticks under standard aseptic methods. The human oral cavity is inhabited by over 600 bacterial species known collectively as oral microbiome. These bacteria are involved in a wide variety of functions and mainly important in maintaining oral health. Oral dysbiosis leads locally to periodontitis, caries, and potentially to head and neck cancer. Cigarette smoke is a source of numerous toxic that come into direct contact with oral bacteria. These toxicants can perturb the microbial ecology of the mouth via antibiotic effects, oxygen deprivation, or other potential mechanism. So these are the materials we use. Methods we use are agar well diffusion method. A cork borer is also used to punch holes on an agar plate and to perform well diffusion assay in microbiology to study bioactivity. Agar well diffusion method is widely used to evaluate the antimicrobial activity of microbial extracts. Similarly to the procedure used in this diffusion method, the agar plate surface is inoculated by spreading a volume of the microbial inoculum over the entire agar surface. For the preparation of sample, we use first the half of the cigarette was burned and the ash was collected in the dilution tube containing saline. For second half, the cigarette was dipped in the dilution tube containing methanol. For other half, the remaining part was kept in the dilution tube containing saline. For sample loading, sample A and sample B are the samples taken from two different humans from their mouth with the help of sterile cotton swab using quadrant method. Well were made in the nutrient agar plate with sterile cork borer. The quadrants were numbered and the samples were loaded in the wells precisely. Results after incubating the nutrient agar plate for 24 hours in the incubator at 37 degrees Celsius zone of inhibition were observed around the wells made in the samples mixed with methanol and saline. We conclude that our normal human mouth microflora is highly affected by the smoke of the cigarette and it has highly pathogenic microbes in it. Thank you.
Now I request Mimansa Tiwari to present her paper to estimate effect of figs on blood glucose level by biometric method.
Hello everyone, my name is Mimansha Tiwari and I am a student of second year microbiology from Thakur Sham Narayan Degree College. My title for the research is Effects of Figs on Blood Glucose Level. This research was done under Mr. Kunal Thakur sir and Mr. Udayban Yadav, sir, from Thakur Sham Narayan Degree College. Starting from the abstract, figs have high nutritional content and have little calories. Abscisic acid, that is ABA, a naturally occurring hormone that controls the body's response to glucose and lowers inflammation, is abundant in figs. Researchers evaluated the extract's ability to reduce blood sugar levels to a glucose solution in different concentrations. The subject was chosen to investigate how figs affect blood sugar levels. In a sugar solution of 200 micrograms per ml distilled water, the enzymatic activity of figs was examined. The medical term for elevated blood sugar level is hyperglycemia. It usually takes high blood sugar levels above 180 to 200 milligrams per deciliter for hyperglycemia to manifest as symptoms. At 530 nanometer, the outcome was then spectrophotometrically verified. A steady decline in descending order was observed as a result. Fixed sugar lowering activity was demonstrated. The common figs come from mulberry family. They are currently cultivated all over the world in temperate areas, but they originate from Western Asia and the Middle East. Figs are inflorescence and are also called syconium. Using the glycemic index, foods are ranked according to how rapidly or slowly they elevate blood sugar levels. The lower a food is on glycemic index scale, the smaller its impact on blood sugar stability. The quicker glucose is released into your bloodstream with a higher food ranking, creating a sharp rise in blood sugar. With a glycemic index of 51, figs fall within the low GI category. Regular consumption can aid heart and intestinal health as well as blood sugar regulation. Figs are low in calories and contain a variety of vitamins and minerals. However, dried figs are high in sugar and calories. The chlorogenic acid present in figs help in reducing blood sugar levels in diabetes. Potassium, which is found in abundance in figs, also play a good role in control of blood sugar levels. They are also a rich source of alkaloids, triterpenoids, vitamin C, and flavonoids. The method used for estimating the blood sugar level in this experiment was DNSA, 3,5 dinitrosalicylic acid. It is easier, convenient, cheaper, and also gives results within 10 minutes. So the materials and methods used for this research was first initially the collection of samples. Fresh samples of fig fruit were collected from nearby market. The edible part was separated and used to determine its enzymatic activity in sugar solution. Then extraction of raw materials. The edible part of the fig is crushed into fine paste using mortar and pestle. Upon adding a little water, it was further strained and filtered using a funnel and a filter paper to remove all clots for smooth and effective procedure. Then preparation of sample. 200 microgram into 20 ml distilled water sugar solution was prepared and 20 ml DNSA reagent was prepared. The requirement table. Since 200 microgram per ml is the concentration of blood sugar in diabetic patients, we have prepared the sugar solution in the same concentration. Then the DNSA reagent was prepared using all the requirements, and the glassware needed for this was clean and dry test tubes and pipettes, and colorimeter at 530 nanometers. By following the procedure step by step, we reached to calculating the OD from the colorimeter at 530 nanometer. Fully plotted with the results obtained from the DNSA test. A gradual decline is seen in descending order. The sugar cell concentration increases by 0.2 ml in each test tube, while equal amount of fig sample was added in all 6 test tubes. The result shows the enzymatic activity of figs in reducing the sugar level. To conclude this experiment, the study and checking of figs' nature of reducing blood sugar level was done. As figs are naturally sweet and also help in lowering the blood sugar level, it is recommended to diabetic patients. In addition to natural sweetness, figs provide fiber and antioxidants. If portion controlled, they can fit into any meal plan. Thank you.
Now I request Tanvi Gowde to present her paper, Comparative Study between Cellulase Enzyme Extracted from Goat and Fish.
Hello everyone, I'm Tanvi Gaudi from ZSCT's Thakur Shamnaran Degree College, working under the guidance of Mr. Kunal Thakur, and our coordinator is Udayban Yadav. My topic name is to determine cellulose activity of organism. Now I'm going to focus on abstract. The biohydrolysis of cellulose through the use of cellulosic microorganism is an attractive approach since the degradation of cellulose by chemical agent produces environmental pollution. Fungal species have been primarily used commercially for cellulase production because of their capacity to secrete cellulosic enzyme into their medium. But however, the bacterial cellulase have several advantages. First, bacteria have higher growth rate than fungi and can easily grow to high cell densities in inexpensive nutrient source. Introduction. Cellulose is an organic compound and polysaccharide consisting linear chain of several hundred to many thousands of glucose units and which is degraded by cellulase uh, with the systematic name 4-beta-D-glucane 4-glucanohydrolase is an any of several enzymes produced by chiefly by fungi and bacteria and protozoans that catalyze cellulosis, the decomposition of cellulose and of some related polysaccharides. Now methods and material. Cellulitic enzyme activity media used in this study were nutrient broth with peptone 0.5 gram yeast extract, 0.2 gram sodium chloride, 0.5 gram and agar 1.5 gram, sodium hydroxide and cellulose extract. To determine cellulose activity, bacteria isolated from intestine of goat and fish were grown in a nutrient broth in a test tube. The test tube were incubated at 37 degrees Celsius for 24 hours. NaOH is added in 1 ml of culture from broth. The samples for cell lysis with the help of Botax machine. This method uses sodium hydroxide to simultaneously extract the lysis lyse cultured cell. NaOH facilitates the lysis by locally changing the pH on a cell, thus facilitating denaturation of cellular compounds. Uh, this is how we isolated the bacteria from intestine of both fish and goat, and the chart according to the enzymatic activity. Now, result and discussion. The comparative study between the goat intestine and fish intestine lead to the conclusion that cellulitic activity of goat's intestine is greater than that of fish. Therefore, this study was conducted to screening, optimization, purification, and characterization of cellulase from cellulase producing bacteria producing in intestine of goldfish and goat. Conclusion. Nowadays, cellulase account for 20% of global enzyme market and they have a biotechnologically potential in various industries. Therefore, the isolation and the screening of cellulitic microbes from various environments are some of the important approaches for obtaining novel cellulase. Most bacteria in nature cannot be isolated and cultivated by traditional culture-based method. The isolation of cellulitic bacteria from unique environment is the challenge for the acquisition of novel cellulase. Freshwater lake provide a unique habitat for diverse bacteria because they differ from other aquatic habitat such as rivers and ocean. This study initially describes the diversity and cellulosic activity of culturable aquatic bacteria from goldfish. Rumen is an excellent environment for the microbial growth and consisting of bacteria, fungi, protozoa, which are widely known to play important role in a fermentation process. The ruminant cattle feed. This is a reference. Thank you.
Now I request Dimple Dubey to present her paper to identify toxic effect of soy sauce and vinegar on human intestinal bacteria.
Greetings to everyone present here. My name is Dimple Dubey and my topic is toxic effect of soy sauce and vinegar on human intestinal bacteria. Introduction about soy sauce. Soy sauce is traditionally used in Japan and several Oriental countries. Is a liquid seasoning currently used in cooking worldwide. The daily consumption of soy sauce in Japan is estimated at about 30 ml per person. According to the data from the Japan Soy Sauce Growers Association, studies suggest that soy sauce contains certain bioactive components in addition to taste and aroma compounds and has various biological functions, including anti-carcinogenic, antimicrobial, antioxidative, and antiplatelet activities, and the inhibition of an angiotensin, a converting enzyme. Therefore, soy sauce is considered to be not only a traditional seasoning, but also a functional food. Soy sauce is probably man's oldest prepared seasoning, processed similarly to miso except that the paste produced is pressed in both Oriental and American cuisine. There are two types of soy sauce, fermented soy sauce and soy sauce made from hydrolyzed vegetable protein, that is HVP. Introduction about vinegar. Vinegar has long been used worldwide as a basic seasoning in the preparation of certain foods because its sharp taste makes it so useful and versatile. A considerable quantity of vinegar is marketed as such as for domestic use. In the UK and USA, the table vinegar is most widely used in cider vinegar, while in Ireland, that is malt vinegar, and in grape growing countries such as Italy, France, and Spain, wine vinegar. In Far East, in addition to the traditional rice vinegar, synthetic vinegar is common. Vinegar adds flavor to vegetable and meat products. It is one the ingredient of salad dressing, sauce, such as Tabasco, and tomato products such as ketchup, mustard, and aspic. Mixed with oil and salt makes the classic vinaigrette, and it can use as a condiment for salad and as a salt for cold, cooked vegetables, meat, and fish. Abstract. Soy sauce is made from fermented paste of soybeans, roasted grain, brine, and Aspergillus oryzae or Aspergillus sorghum. To avoid microbial contamination during the production process, 16 to 20% weight per volume of sodium chloride is commonly added. High salt food causes health risks such as high blood pressure, heart, and kidney disease. Therefore, the sodium chloride content of soy sauce should be reduced to 5 to 8%. Studies have shown that the soybeans fermented by microorganisms also provide a variety of functions, such as antioxidation, lowering blood pressure, lowering blood sugar, anti-cancer, anti-aging, etc. Others suggest vinegar slows the rate of gastric emptying, which would slow the rate of glucose absorption into the bloodstream. It has been suggested that accidental inhalation of vinegar can cause laryngospasm. Vinegar can cause erosion of dental enamel. Vinegar can damage the esophagus, hypokalemia, hyporenemia, and osteoporosis have been attributed to the ingestion of a large amount of cider vinegar. Materials and methods. Firstly, prepare 2 ml suspension for bacteria, Staphylococcus aureus using saline, and take Nutrin agar paste with 1 ml of bacterial suspension. Prepare on Nutrin agar paste with the help of sterile pipette. Spread it on Nutrin agar paste using a spreader. Incubate the plate at room temperature for 10 minutes. After that, make wells in agar using corporate with the sample of soy sauce in one well and put the sample vinegar in another well with the help of microwave. Incubate the plate at 37 degrees Celsius for 24 hours. All these procedures should take place in aseptic zone condition to make sure there's no contamination in a agar plate. All the equipment, materials used to be sterile. while putting agar plate in incubator, do not invert the plate. Results. After incubation of 24 hours, results are checked. We found a zone of inhibition for both the samples of soy sauce and vinegar. Zone of inhibition for vinegar is more environmental than the soy sauce. That means vinegar is more toxic for the bacteria present in the human intestine. Here we can see the results for the soy sauce and the vinegar. The plot 1 is for the vinegar and plot 3 is for the soy sauce. Conclusion. Both soy sauce and vinegar have benefits and side effects also. It affects bacteria present in the human intestine. Today, soy sauce and vinegar both are used in huge amounts in almost many Asian countries, so it is important to know its effects. We can conclude that both the samples are harmful on bacteria, Staphylococcus aureus, because zone of inhibition was in results. Thank you to my respected teachers and principal and everyone present here. Thank you so much.
Now I request Asmi Save to present her paper, that is estimation of antimicrobial properties of shrimp extracted on E. coli.
Greetings to everyone. My topic is estimation of antimicrobial property of shrimp extract on E. coli. This was performed by me, Ms. Asmi Save, under the guidance of Mr. Udayban Yadav sir and Mr. Kunal Thakur sir. Submitted to the Department of Microbiology ID, ZSCT Thakur Shamnaran Degree College, Mumbai University. In this research, we tested the antimicrobial activities of sample mentioned below. The specimen utilized was shrimp extract from the shrimp waste. Shrimps are found near the seafloor along most coast, estuaries, river, and lake. Mostly they are found in marine water, but they are also found in the fresh water. Marine species can be found from tropic to the pole at a depth of up to 5,000 meters. Shrimp are eaten by a variety of animals like fish or the seabirds. They also play a vital role in food chain and provide food for large animals ranging from fish to whales. Shrimp waste, especially the dry head or shell, was discovered to have a high protein content as well as high level of mineral, particularly calcium, phosphorus, sodium, and zinc. Shrimp contain chemical components like pigment, astaxanthin, and chitin, and also used to get antibiotics as given. The research examined the antimicrobial effect of shrimp extract against Escherichia coli. In this experiment, we employed the agar-well method to load sample. After 24 hours of incubation, we saw a small zone of inhibition. Materials and methodology. The methodology used is agar-well method. Materials. Shrimp sample crushed in mortar and pestle. 1 petri dish, McConkey agar, cork borer, pipette of 1 ml and 10 ml, saline suspension of E. coli, spreader, distilled water, nichrome loop, methanol. Sample was prepared by extracting shrimp from the shrimp waste by crushing it in the mortar and pestle and adding methanol. This methanol was later on evaporated and the sample was put in distilled water. The procedure used was agar well method. Sample was loaded in the well made in the center and incubated for 24 hours. On next day, plate were observed and the diameter of inhibition was measured. As we see a slight zone of inhibition in the given photos. After incubation, a zone of inhibition was observed around the well of the sample. 19 mm zone of inhibition was observed. Shrimp has been the best part of our diet of many humans for years. As we know, it consists of high level of protein, minerals, and also pigments. Shrimp also show antimicrobial activity, and various antibiotics have been extracted from shrimp according to the reference given. After performing the experiment, we observe a zone of inhibition concluding that shrimp shows antimicrobial property against E. coli. Thank you.
Now I request Pratham Das to present his paper, Estimation of Antibacterial Property of Athers against Normal Skin Microflora.
Hello, I am Mr. Pratham Das from ZSCT's Thakur Shyamnarayan Degree College, and my today's research topic is estimation of antibacterial properties of attar. Under the guidance of Mr. Kunal Thakur sir, Assistant Professor, and Mr. Udayban Yadav, Coordinator of Microbiology Department. Moving forward with the topic, a herbal green opportunity of perfumes is other that are non-alcoholic oil primarily based totally herbal extracts derived from the flowers, herbs, woods, etc. Other is an aromatic crucial oil. It has exhilarating and spiritually raising impact. Aroma other contains crucial oils that have a calming, soothing, and enjoyable impact. It's also recognized to offer comfort from headache. Other additionally have medicinal values. It can assist in comfort from asthma, cough, and different breathing problems. Escherichia coli, Klebsiella pneumoniae, Staphylococcus aureus are the predominant organisms found on the skin. Skin microorganisms have essential role in protection against invading pathogens, the education of our immune system, and the breakdown of natural products. Other may affect the normal flora of the skin because of the presence of triglycerides. The zone of inhibition is a circular area around the spot of the antibiotic in which the bacterial colonies do not grow. A zone of inhibition test, also called a cork borer test, is a qualitative method used clinically to measure the resistance and industrially to test the ability of solids and textiles to inhibit microbial growth. By using this technique, by using this test, the effects of other on normal skin flora can be studied. Introduction. The word attar or ether comes from the associated historic Persian word attar, otto, or otter, which means fragrance. Perfume or heady fragrance producing attar is the exercise of extracting heady fragrance from the flower, herbs, and different botanical sources. Some essential oils are extracted from the timber species as nicely. Attar is an essential oil derived from biology and alternative natural resources. The oils are generally distilled in a wood base resembling wood, then aged. The aging amount can last from 1 to 10 years, counting on the botanical use and therefore the results desired. Others are various types extracted from various types of flowers and fruits, peels. Some common types of attar are mogra, orange, rose, lemon. The skin is the human body's largest organ, settled by a various environment of microorganisms. Most of that are harmless, could be perhaps helpful to their host. Skin microbiota plays associate tangled role within the human system. Really several immune functions and helps to defend its host against destructive microorganism pathogens. Methods and materials. Sample collection. The sample of normal skin flora through cotton swab was taken in sterile saline test tube. The ether of 4 different aroma were selected: mogra, lemon, orange, rose. Isolation of sample on nutrient medium. The sample collected was plated on nutrient agar plate through spread plate technique using a sterile swab and incubated for 24 hours at 37 degrees Celsius. 3 isolated organisms were found on the nutrient agar plate after incubation: Escherichia coli, Klebsiella pneumoniae, and Staphylococcus aureus. All the 3 organisms were plated on 3 different nutrient agar plates to the spread plate technique and kept at a room temperature for 30 minutes. Agar well diffusion method. 3 holes, also called wells, were made on each of the plate and the ether sample was diffused into the wells. The plate were incubated at 37 degrees Celsius for 48 hours to observe the zone of inhibition. These are the pictorial representation of the plates. Figure 1 representing Escherichia coli and figure 2 representing Staphylococcus aureus. The figure 3 represents Klebsiella pneumoniae. This is the observation table showing the inhibitory zone diameters. These are the bar graphs showing antibacterial growth identification. These are the pie charts of the same. Result. After inoculation, zone of inhibition were observed around the wells of each sample. Lemon and orange showed visible zone of inhibition in all three plates under the used bacteria Escherichia coli. Staphylococcus aureus and Klebsiella pneumoniae, rose, and mogra showed minimal zone of inhibition. Conclusion. This experiment concluded stating that naturally available ether like rose, mogra, lemon, orange possesses antibacterial properties which can be effective against normal skin flora, bacteria like Klebsiella pneumoniae, Escherichia coli, and Staphylococcus aureus. These substances will be helpful in increasing our immunity and fight against in many infectious bacteria on the skin. As these are having organic sources, there will not be any side effect which can be seen in case of antibiotics. Finally, according to my research, these substances are useful. This is the bibliography. These are the references. Thank you.
Now I request Nidhi Mane to present her paper Estimation of Antibacterial Property in Ginger Garlic and Ginger Garlic Paste Against Food Poisoning Causing Bacteria.
Hello, I'm Ms. Nidhi Mane from City's Thakur Shamnara and Degree College, and my research topic is estimation of antibacterial properties in ginger, garlic, and ginger garlic paste against food poison causing bacteria under the guidance of Mr. Kunal Thakur, Assistant Professor of City's Thakur Shamnara and Degree College, and Mr. Udayban Yadav Sir, Coordinator of Department of Microbiology. Moving forward with the topic, many of the species used in our daily diets have been noted to have great medicinal values and antibacterial properties as well. Ginger and garlic are one of these spices, which are very useful and important and are used as therapeutic agent against many infections. The aim of this research paper was to estimate the antibacterial properties found in ginger, garlic, and their paste against three strains of food poisoning bacteria, such as Pseudomonas, Klebsiella, Escherichia coli. The method that was used for this was corporal well diffusion method. The samples were loaded by 1% volume per volume dilution in the well following the incubation of 24 hours. The growth was observed, which evidently showed that garlic has the most antibacterial properties against all three bacteria, whereas ginger and ginger garlic paste have minimum antibacterial properties against this bacteria. Food poisoning, also called foodborne illness, is an infection that spreads through foods and drink. Food poisoning or foodborne diseases is one of the major problems in public health worldwide. Organisms such as Pseudomonas, Klebsiella, Clostridium, Perfringens, Camelobacter, Bacillus cereus, and Escherichia coli are common food poisoning agents. Commonly, prevention of food spoilage is achieved by the use of chemicals, preservatives, but repeated application of these chemical preservatives may result in some negative effects such as accumulation of chemical residues in food, acquisition of microbial resistance to the applied chemicals, and unpleasant side effect of these chemical preservatives on human health. Talking about the materials and methods. Preparation of samples. Samples were prepared using two, that is ginger and garlic. Ginger and garlic were first finely crushed, making a thick paste of it. Then the three samples were prepared from it, which included ginger paste, garlic paste, and one, mixing both of them together, forming a ginger garlic paste, after which 1 gram of each sample were diluted in 10 ml of saline solution to form a semi-liquid consistency. Inoculation of bacteria. Klebsiella, Pseudomonas, and Escherichia coli were isolated and inoculated in saline solution and kept in incubator for 1 hour to observe a mat growth. After incubation, the saline suspension was pipetted out from a sterile 10 ml pipette and loaded in agar plates. The bacteria was then spread equally to all sides using a sterile spreader. Sample loading. Using quadrant method, wells were made in the agar plate with a sterile cork border. The quadrants were numbered and the samples were loaded into the wells precisely. Observing the growth, the agar plates were kept in incubation for 24 hours. After incubation, it was observed that garlic showed visible zone of inhibition against all bacteria, whereas ginger and ginger garlic paste also showed very minimal zone of inhibition against the used bacteria. These are the photos of the organisms that showed zone of inhibition. First is Klebsiella, second is Pseudomonas. This is Escherichia coli. This is the observation table which shows the diameter of the zone of inhibition in mm, which interprets that garlic showed the most zone of inhibition and ginger garlic paste showed minimal zone of inhibition. This is the bar diagram which shows antibacterial growth identification of all the samples. The pie chart for each bacteria which was grown in ginger, garlic and ginger garlic paste. Result. After incubation, zone of inhibition were observed around the wells of each sample. Garlic showed visible zone of inhibition in all three agar plates under the used bacteria, that is Klebsiella, Escherichia coli and Pseudomonas. Ginger and ginger paste showed minimal zone of inhibition. For concluding this paper, this experiment concluded that naturally available spices like ginger and garlic possesses antibacterial properties which can be effective against food poison causing bacteria like Klebsiella, Escherichia coli and Pseudomonas. So we can use this to prepare natural sanitizers also. The substances will be helpful in increasing our immunity and fight against many infectious bacteria. As these are having organic sources, there will not be any side effect which can be seen in case of antibiotics. Finally, according to my research, these substances are useful. This is the bibliography for the research paper and the references. Thank you.
Now I request Manisha Thakur to present her paper to check the toxicity of plaster of Paris on water's natural microflora.
Hello everyone, myself Manisha Thakur from ZSCT's Thakur Shamnarayan Degree College, Mumbai University, Department of Microbiology. I'm working under the guidance of Mr. Kunal Thakur, Assistant Professor in Thakur Shamnarayan Degree College. So my topic is to check the toxicity of the plaster of Paris on natural waters microflora during Ganesh Chaturthi. We'll go to the abstract of it. Ganpati idol immersion each year during Ganesh Chaturthi is adding a load of pollution to natural water bodies and microflora of these water bodies too. Non-biodegradable materials and synthetic paint used in the making of these idols causes a serious threat to the microflora because of their chemical contents and that may resist the growth of the bacteria. Present study was conducted to observe the effect of idol made up of POP, that is plaster of Paris, on water's natural microflora. For this, the water sample was collected after the immersion of Ganpati idols from Sanjay Gandhi National Park. The water had a great amount of calcium. There was a change in pH of water. Hardness was seen in the water taken from the immersion. After the immersion, hydrological changes were observed in water due to Ganpati idol immersions during and after the Visarjan. Introduction. The most vital resource for life is water. There cannot be life without fresh water. The issue of water are becoming increasingly important to environment, particularly to the human health and the food. Festivities such as Ganesh Chaturthi pollute the water and make it hazardous for human as well as natural microflora of the water. It is important that there should be a less use of Ganpati idols, anything that is made up of POP, clay, or which is not biodegradable in water and may cause pollution to the water and may resist the growth of natural microflora of the water. Now we will see how we perform the experiment overall, the procedure and methods and materials used in it. The present study was carried out in the laboratory to find out the impact of plaster of Paris idols of Ganpati. A water containing POP after Ganpati Visarjan was taken to study the impact of these idols on the natural microflora of the water. Collection of samples. The water sample containing plaster of Paris was taken from natural pond of Sanjay Gandhi National Park. After the Ganpati Visarjan, the color of this water sample was dusty white. The water sample containing POP was stored in plastic container and capped and labeled properly. Then the sample was taken in for laboratory analysis. Bacterial stains that we used to perform these experiments were culture suspension of Pseudomonas aeruginosa and Escherichia coli. Isolation of the bacteria. Uh, Around 1 loopful culture of Pseudomonas aeruginosa and Escherichia coli was suspended in 10 ml of saline solution, after which 2 ml was inculcated upon the Petri dish using spread plate method. After the spreading, incubation for 30 minutes was done. We made one well in both of the sterile plates containing agar with coke with sterile coke borer, after which 0.1 ml of water sample was added to the well with the help of micropipette. Petri plates were incubated at 37 degrees Celsius for 24 hours. Screening by coke borer method or well diffusion agar method. Antimicrobial activity of self-free extract was evaluated by coke borer method against the indicated organism. A lawn of bacteria was expected to grow after the spreading of the suspension of the culture on the surface of agar. Sample of water containing POP was incorporated on both the wells of the respective plates at an amount of 0.1 ml and were incubated for 24 hours at 37 degrees Celsius. The result was recorded by observing and measuring the zone of inhibition. In result, a zone of inhibition was not observed on any of the plates. This does not necessarily indicate that microorganisms have been killed or that the POP resists the growth of it. Conclusion. After the 24-hour incubation of Petri plates under lab conditions, it was concluded that presence of plaster of Paris in the form of idle does not inhibit the growth of natural and normal microflora of the water, but it does not dissolve in water as well. So it may cause the problem of sedimentation and cause pollution in the water. Thank you. This is the reference paper.
Now request Viral Patel to present his paper, Change in Mouth Diversity After Food Habitats.
Hello everyone, my name is Viral Patel. My topic for research is changes in mouth diversity after food habitat. And I am doing this research under my Professor Mr. Udayan Yadav. In recent decades, a body of literature examining the relationship between health and general health has rapidly developed. However, the biological mechanism only explaining such relationships has not been fully described. Recent evidence has suggested that these relationships could be partially explained by the composition and interaction of the microbiome microbiota between local and the cemented body sites. For instance, it has been suggested that the intestinal microbiota would have effects on non-communicable diseases such as diabetes or cardiovascular diseases. The objective of this study is to explore current evidences of the link between oral and semantic diseases, discuss whether oral microbiome or biota could represent an unexplored biological pathway, partially explaining those relationships. A non-semantic review of this literature was carried out using keyword research in from February to May 2019. The ultimate goal was to present recent scientific evidences to update the general knowledge on this topic to professional and dentist practicing. This review is divided into two parts for journal publication. However, it is intended to be used as one piece. In the first part, we will summarize the conceptual background of oral microbiome microbiota. We will describe the main methods used in the microbiology to characterize oral microorganisms. I will present the main composition of bacteria in oral microbiome microbiota. Introduction. Oral microbiome is defined as a collective genome of microorganisms that resides in the oral cavity. After the gut, it is the second largest microbiome community in the humans. As compared with the other body sites, they exhibit an astonishing diversity of predicted protein functions. Human microbiome consists of a core microbiome and a viable community. The core microbiome is common to all the individuals, whereas the variable microbiome is unique to individuals depending on the lifestyle and the physiological differences. The oral cavity has two types of surfaces on which bacteria can colonize: the hard and the soft tissue of the teeth and the oral, mucosal, respectively. The teeth, tongue, cheeks, hard plates, and soft plates provide a rich environment in which microorganisms can flourish. The surface of the oral cavity are coated with a ladder of these bacteria. The flowable bacteria biofilms. An ideal environment is provided by the oral cavity and associated with nasopharyngeal region. For the growth of the microorganisms, the normal temperature of the oral cavity on an average is 37 degrees Celsius without significant changes, which provides the bacteria a stable environment to survive. Saliva also has a stable pH of 6.5 to 7, several pH for the most species of bacteria. It keeps the bacteria hydrated and also survives as a medium for the transportation of nutrients to microorganisms. Materials and methods. Materials used in this experiment are sugarcane, nuts, lemons. Sterile nutrient media, sterile swab, dye crystal boiled, methylene bread, alcohol, safranin, and microscope. Method, gram staining, spreading, and streaking. Suspension of all the three food samples were prepared using saline. Nutrient plates was used for the isolation of microorganisms. Isolation of airmark microfauna was done by using isolation method. For this nutrient plates were used for preparation of nutrient agar. 28 grams of nutrient agar was added to 100 ml of distilled water, and the medium was sterile at 120 degrees Celsius at 15 psi pressure. 20 ml of sterile nutrient agar was poured into the petri plates, and medium was allowed to cool till it is solidified. The results of the isolated colonies are here, which were incubated for 24 hours. Other characteristics: shape, concave, size 1 to 3 mm, elevation convex, surface smooth, stress isolation rough, repeated subculture, mucoid, capsulated strains. Color grayish white, structure translucent opaque, amplificability smooth forms, easily emulsifiable, rough form, autoaccumulable, hence do not amplify easily. Results of gram staining. Result: bacteria that can grow in acidic condition are Acidobacter bacterium, Acidobacillus febreoxidans, Pedicom, Cocum acidilactic. Bacteria that can grow in basic condition: Bacillus, Pseudomonas, Streptomyces, and Sinusitis. Bacteria that can grow in neutral condition: Escherichia coli, Streptococci, and Salmonella. Conclusion: the oral microbiome is an exciting and expanding field of research. Oral microbiome is curricular to health as it can cause both oral and semantic diseases. It rests within the biofilm throughout the oral cavity and forms an ecosystem that maintains the health in a state of equilibrium. However, certain imbalance in the state of equilibrium allows the pathogens to manifest and cause diseases. Disruption of the microbiome leads to dysbiosis identification. The microbiome in health is the first step of human microbiome research, after which it is necessary to understand the role of the microbiome in the alteration of the function of and metabolic pathway associated with the disease states. Microbiome research is currently at a very nascent stage. A lot of research is being done and the data are added continuously. However, the results obtained from the various studies are not consistent. This may be due to the techniques used and the standardization of method sample size. Studies with a large sample size involved different sites in the health and disease are required, which may develop consistent patterns to generate confident data. This will further identify different biomarkers and assist in targeted therapies and personalized medicines for better patient management in the clinical practice. Thank you.
Present her paper, Recent Approaches Towards Various Copper Remediation.
Good day, everyone. Myself, Seenu Maya. I've conducted a research under the guidance of Udayban Yadav sir. My topic is recent approaches toward various copper remediation. References. Introduction. We performed experiment to find vector starting and sanitizing properties of copper and copper alloy surfaces, which demonstrated that copper in very small quantity has the copper alloy surfaces have intrinsic property to destroy a wide range of microorganisms. Today, copper in the form of plumbing tube copper or copper alloy surfaces proved to be a significant step in decreasing the fungal and bacterial insecticides in hospitals or infections. To know the bactericidal and fungicidal properties of copper for its implication in various areas in prevention, bacteriostasis, and sanitization property of copper, which demonstrated that copper in very small quantities has the copper alloy surfaces have intrinsic property to destroy a wide range of organisms. To know the bacterial and fungicidal properties of copper for various implications in various areas preventing nosocomial infection. Metal ions, either alone or in complexes, have been used for centuries to disinfect fluids, solids, and tissues. In Greek era, they used copper as sanitization material for many diseases or in medical conditions. Mechanism of antibacterial action of copper. The antibacterial of copper are still under active investigation. Molecular mechanism responsible for the antibacterial action of copper have been a subject of intensive research. Scientists have actively demonstrated some of the molecular mechanisms mentioned by researchers are the copper alternative, the three-dimensional structure of protein and distribute the enzyme structure, resulting in inactivation of bacteria or viruses. Copper produces deleteration effect in superoxide radicals, generating OH radical, thereby causing multiple hit damage at target tissues. Copper with lipids produces holes in the cell membrane, damaging the integrity of cell and leaking of essential nutrients, leading to cell death. Many recent publications also showed that microorganisms are rapidly killed on metallic copper surfaces by contact killing mechanism. The earliest medical texts refer to use of copper compounds for wound healing, such as sterilization of wounds. Review of literatures. Material and methods. Material are mainly used are in the media of copper substance and different concentration of copper. It is mainly an observational study and conducted in Thakur Shamnarayan Degree College, Mumbai University in microbiology department. Specimens were E. coli organism of pure growth, different concentration of copper water and copper wire, Wattsman paper, nutrient agar media, normal water and boiled water. Media was prepared by dipping the Wattsman paper in water of different concentration, and the E. coli was inoculated on the broth. At different concentration were boiled and kept on normal temperature for different time. It was observed that the growth was observed of E. coli, and on which the copper concentration was placed, there was no growth or less growth. Conclusion. The antibacterial properties of copper surfaces have now been firmly established. Hospital trials have shown a reduction in bacterial counts, indicating that copper surfaces are really additional tool along with other hygienic measures to decrease the number and severity of nosocomial infection. Additional studies should be done in determining the most cost-effective way to protect hospitals so that different sites like doorknobs, bed lids are all made of copper to avoid infection. Thank you.
Now I request Sakshi Singh to present her paper, Isolation of Identification of Microflora in Mangroves Area.
Hello everyone, myself Sakshi Singh. I am working under the guidance of Mr. Udayban Yadav sir. My research topic is isolation and identification of microflora from mangrove areas. Abstract. Covering a quarter of the world's tropically coastlines and being on of the most threatened ecosystems, mangroves are among the major source of terrestrial organic matter to oceans and harbour a wide microbial diversity. In order to productively restore and better understand these ecosystems, researchers have extensively studied their microbiology, yet few surveys have focused on their fungal communities. Our study indicated the presence of survival species on this microbiome that were not previously reported as a mangrove associate. Keywords: microflora, air microflora, microbial study, etc. Introductions. The mangrove area accounts for the 0.7% of total tropical forest of the world. The largest extent of the mangroves is found in Asia 42%, followed by the Africa 20%, North and Central America 15%, Oceania 12%, and South America 11%. Mangrove forests organized in Southeast Asia, but they are consistently under created and destroyed to make way for road and buildings, commercially, aquaculture, and by marine populations. Mangrove distribution in India in 2005 data of survey of India show an extent of 4,445 square kilometer mangrove areas in India. Out of the total mangrove areas, acres 57% of the found on the east coast, 23% on the west coast, and the remaining 20% on the Nicobar Islands. This is a pie chart regarding to the total mangroves in India. Mangroves variations. The mangrove flora of the world is represented by about 65 species. If the vivipary and breathing roots were taken into consideration, there would be 55 species in the world. The floral diversity of mangroves in India is great. In India, mangroves are represented by the approximately 59 species. From 29 families of the 59 species, 34 species belong to 21 families are present along the west coast, and 48 species belong to 32 families are present along the east coast. Mangroves animals having the specialized root system and other morphological adaptation mangroves from dense forest on the shorelines, creating a spread habitat for a variety of fauna. Methods and materials. Isolation of microflora was done by using the soil sample from the mangrove areas for this nutrient agar plate, and potato dextrose agar plate was used. Methods. Soil samples were collected from the different locations and brought to the laboratory for further detection. The suspension of the soil sample were made and hence extract using the nichrome loop on the nutrient agar plates and potato dextrose agar plates under the aseptic conditions. The plates were kept for incubation at 37 degrees Celsius for 24 hours, and results were observed the next day. The isolated colonies were observed and hence suspension was made for gram staining. The gram stain slides was observed under the microscope and the fungi species was detected. Result and discussion. This was the accordance with the research of Ulema that there were bacteria genus found in sediments in the mangrove, namely Bacillus, Enterobacteria, Listeria, Micrococcus. That the diversity of sedimentary microorganism communities was very high. This is due to the connect of sediments, supporting and assisting in the formation of aerobic and anaerobic microenvironment cooperation. Conclusion: mangrove regions are a unique semi-region with water region being alkaline in nature and sediment or soil region having or natural to slightly acidic pH. Since mangrove environment is prevalent to stress condition such as a salt stress, microorganism growing under such stress condition could have a potential for bioremediations programs. The soil isolates their hallow-tolerant and could tolerate relatively high concentration of heavy metals. Mangroves are saline, coastal ecosystem rich in carbon and other nutrients. The harbor large numbers of population of unique bacteria. The present study reveals the mixed population of bacteria of Gorai mangroves area. Further studies on these microorganisms and more evaluation of their stress tolerance could make them applicable for virus industrial applications. Thank you.
Now I request Shivam Kumar Kanochia to present his paper Phytoextraction of Essential Oil used as a natural preservatives for creams and gels.
Hello everyone, my name is Shivam Kumar. I am working under guidance of Mr. Udavan Yadav. My title is Phyto Extraction of Essential Oil used as a natural preservative for cream and gel. Abstract. Skincare is the main part of the appearance of our daily routine. We use cosmetic which most of them are synthetic. Many herbal product industry use the synthetic material as a preservative like methylparaben, paraben, betylparaben, which are preserve cosmetic product long life. The plant is having antibacterial, antifungal, antioxidant, anti-inflammatory properties. Many plant extract species in essential oil having property to kill the microorganism or stop their growth and preservative cosmetic product long life. Introduction. Natural skin care is the care of the skin using naturally derived ingredient such as a herbal root, essential oil, and flower combined with a natural occurring carrier agent, preservatives, effective, emetic, and everything from natural soap to oil to pure water. A classic definition of a natural skincare is based on using botanical sources ingredient currently accepted and formed by nature without the use of synthetic chemical and manufactured in such a way to preserve integrated and ingredient. Material and method. The experiment material get gel and cream, essential oil used as a preservative collected from the Gayatri Herbal Private Limited. Composition base aloe gel 100 ml, 88 ml spring water, xanthan gum 10 ml aloe vera extract. Method. Measure the weight of junk food into bowl. Well, 1 gram of measure level 2 ml measuring spoon and xanthan gum powder. After boiling the sparing water in the candle, measuring the according to recipe and pour it over the emulsion. Glycerin cerate preservative which are put into a separated double boiler. When both fat and water stage are over 715, remove both double boiler from the hob, keeping the water state mixture hot by leaving it at top half of double boiler. Result and dimension. Lavandula officinalis and Rosmarinus officinal to be used as a natural cosmetic preservative in aqueous cream formulation for antimicrobial activity against bacterial and fungi. All the test microorganisms used in the study were general mode susceptibility of the oil during the challenge test in aqueous cream compounds to antimicrobial tests performed on agar. Conclusion. In this study, the herbal cosmetic products were tested for different test firmament. Two herbal cosmetics selected at a regular interval, microbial contamin, were also studied to prove efficiency of linseed oil as a natural preservative and exhibited excellence. Antibacterial, antifungal properties, those natural preservative where does not change in the pH, moisture content, texture, stability, and herbal cosmetic product. Tea tree oil will were so more effective result as a natural preservative compared to eucalyptus oil in gel. Tea tree oil were so more effective result as a natural preservative as compared to eucalyptus oil in cream. Thank you.
Now I request Sivanand Yadav to present his paper, Study of Mycotoxin from Infected Jawar and Bajra by using fungus aspergillus.
Hello everyone, my name is Sivanand Yadav. I am working under guidance of Mr. Udayban Yadav. My title is Study of Mycotoxin from Injected Jawar and Bajra by using fungus and aspergillus. Abstracts. Natural toxins in food are important part of our life. As per oxygen we breathe, it is very important naturally neutralize these toxins. Our diet order the consume food safety. Not only would be impractical, but would be also impossible to completely eliminate natural toxins from the diet. Fungus and bacteria are main causes food poison and symptom the diarrhea, stomach and fever will be developed in about 3 days. Introduction. Mycotoxins are secondary metabolism produced by fungus diet. Their pharmacological activity. Some mycotoxin and mycotoxin derived have been used to antibiotic growth, promotes growth, and the considered chemical welfare agent. They are obtained name after the fungus and produce them. For example, alpha toxin used in letter A, aspergillus plant and flea species with word toxin. There are three main mycotoxins produced by fungus: aspergillus, Rickettsia, and penicillium. Mycotoxin produce fungus growth and food inducing grain, bean and pea. Material requirement: potato turmeric 100 gram, dioxy 10 gram, agar 7.5 gram, peptone 1 gram, dilute water 500 ml, SL 1 number, NaOH 1 number. Procedure: take a potato peels and weight 100 gram. Use the knife cut and trim a small species. Transfer the coated potato cup connecting about 200 ml dilute water, etc. Finally, transfer the medium to 500 ml granted cylinder and up to 5 ml by adding the dilute water. Measuring pH medium and adjust to 5.6 with 1 N SCl and NaOH. Pour the medium into the oil seal bottle, plug and cotton stopper and covered with aluminum foil. Once the temperature has been.
Now I request Mayur Patil to present his paper, Study of Microflora in Mangroves Area.
Warm regards to one and all present. Myself, Mayur Patil. Today, I'm going to give my presentation on the topic study of microflora in the mangrove region. Abstract. Bacteria plays a major role in the food chain and water productivity. Mangrove ecosystem provide important ecological benefits and ecosystem services, including carbon storage and coastal stabilization, but also suffers from major anthropogenic pressure. Understanding this connection and moving from the descriptive studies of microbial taxonomy to hypothesis-driven field and laboratory studies will facilitate mechanistic understanding of mangrove ecosystem, interaction, network, and open opportunities. Materials and method. Suspension of all three soil samples were prepared using saline. Nutrient agar plates were used for the isolation of microorganisms. For preparation, nutrient agar. 28 grams of nutrient agar was added to 1 liter distilled water and the medium was sterilized at 120 degrees Celsius at 15 lbps pressure. 20 ml of sterilized nutrient agar was poured into sterile Petri plates and medium was allowed to cool it till it solidified. Samples were collected from the location of Gorai Mangroves area. The samples were collected from the low, medium, middle, and high tide, inoculated with saline, and prepared a suspension with the help of micron wire inscribed on nutrient agar plate. Different nutrient agar plate was used and labeled as low, middle, and high tide. It was performed under aseptic zone and kept it in the incubation for 24 to 48 hours for 37 degrees Celsius. Result and discussion that there were bacterial genus found in sediments in the mangroves, namely Bacillus, Enterobacteria, Listeria, and Micrococcus. Bassett et al. Stated that diversity of sedimentary microorganism communities were high. Due to this, of the content of sediment supporting and assisting in the formation of aerobic and anaerobic microorganism, microenvironment cooperation. For example, a decrease in oxygen level due to microorganism activity in a room rich in organic matter will form an anaerobic microenvironment that supports facilitative and obligate anaerobic microorganism activity. This causes the emergence of groups of microorganisms with certain specific physiological properties that are in accordance with the condition of microenvironment. After staining it, the colony characters were rod shaped and gram-positive when cultured on ordinary nutrient agar. The morphology circular colonies of this bacteria is rough, opaque, fuzzy, white, or slightly yellow with jagged edges. The bacteria was Bacillus, bluish-gray in color, with a shiny texture. It often looks like mucus or cloudy film over whole surface of the plate. Colony is slightly raised and has an entire fixed margin and a steady growth pattern, creating concentrated growth rate in the colony. The bacteria was Enterobacter. Yellow color colonies have appear rough, smooth, convex, uh, nicely with edge, entire edge. And the bacteria was micrococcus. Gray to off-white color appeared colonies have a low convex profile on nutrient agar. Colonies appeared bluish-gray by normal illumination. The bacteria was Listeria. Conclusion: the mangroves region are unique swampy region with the water region being alkaline in nature and sediment or soil region having a neutral to slightly acidic pH. The soil isolates were halotolerant and could tolerate relatively high concentration of heavy metals. They harbor large number of population of unique bacteria. The present study reveals the mixed population of bacteria of Gorai mangroves area. Now, I would like to conclude my presentation and thanks our professor, Mr. Udayvan Yadav, giving us this opportunity and guiding throughout. Thank you.
Now I request Karina Jaiswal to present her paper In vitro antifungal activity of Argemon mexicana against two fungus, Aspergillus niger and Candida albicans.
Warm regards to one and all present. Myself, Karina Jaiswal. Today I'm going to give my presentation on the topic in vitro fungal activity of bioactive compounds from Argemon mexicana against two fungus which are Aspergillus niger and Candida albicans. Abstract. Fungi are indirectly to be blamed for allergic or cytotoxic disorders and the assembly of mycotoxins or allergens. Generally, for the management of phytopathogenic fungi, use of artificial fungicides is finished. These chemical fungicides have tremendous effect on the plant as well as on the animals feeding on them. Hence, extracts of oils of medicative plants have been used since it contains a great deal of secondary metabolites as compared to other plants. Materials and method. In materials, two types of fungi were used, Aspergillus niger and Candida albicans. The plant used was Argemon mexicana, which has its antifungal property. Two types of culture were used, liquid culture medium, which is the potato dextrose broth, and the solid culture medium, which is the potato dextrose agar. Other requirements were also used, and this entire experiment was carried out in aseptic conditions with sterilized glasswares. Method. Firstly, the aqueous extract of Argemon mexicana was prepared of 5 different concentrations ranging from 10 to 50%. Then, the plant aqueous extracts of Argemon mexicana was tested in both the mediums by inoculating the fungi in it. This table shows the volume of extract and volume of broth used. After 7 days of incubation, the fungal growth was observed. The biomass was prepared by drying the filtrate in the oven at 180 degrees Celsius for 2 hours. On solid culture medium, the antifungal activity of solid medium was studied using agar well diffusion method. A controlled Petri plate was maintained without extract for comparison, and the Petri plates were incubated for 7 days to observe the zone of inhibition. Observation and result. In liquid culture medium, the Argemon mexicana extract on Aspergillus niger. It showed excellent growth at 10% and it showed inhibition at 50%. Activity of Argemon mexicana on Candida. It also showed inhibition at 50% concentration. On solid culture medium, the zone of inhibition of Argemon mexicana Candida was the largest at 50% of concentration. Result. On liquid culture medium, the growth of Aspergillus niger was completely inhibited at 50% concentration and other concentrations showed fair amount of growth. The growth was very slow for Candida on aqueous extract and it showed MIC at 50%. On solid culture medium, Aspergillus niger was used to be resistant on the plant extracts. The agar well diffusion method showed a clear zone of inhibition on higher concentrations of the extracts of Candida. For aqueous extracts for Argemon mexicana, as compared to the control plate and other concentrations, Candida albicans showed a clear zone of inhibition on 50% concentration. Conclusion. The above experiment has concluded that the aqueous plant extract of Argemon mexicana is effective against both the pathogenic fungi Aspergillus niger and Candida albicans. Aspergillus niger showed more resistant at more concentrations, Candida being sensitive to the antifungal compounds present in the extract shows considerable decrease in biomass and showed complete inhibition at the highest concentration, 50%. Studies indicate that the higher the concentration of plant, higher is the effect of the inhibition. With this, I would like to conclude my presentation and really like to thank our professor, Mr. Udayban Yadav, for giving us the opportunity and guiding us throughout. Thank you.
I request Lalit Rawat to present his paper in vitro antifungal activity of Xanthium stomachum freshwater plant extracted against two fungus which are Aspergillus niger and Fusarium oxysporum.
Warm regards to one and all present. Myself Lalit Rawat. Today I am going to give my presentation on the topic in vitro fungal activity of Xanthium stomarium fresh plant extract against two fungus which are Aspergillus niger and Fusarium oxysporum. Abstract. The soil is a home to a wide range of microorganisms including fungus, bacteria and viruses. Few of those microorganisms are beneficial to the soil, but many of them have the potential to seriously harm plant development and degrade soil quality. Healthy plant development will result from preventative actions made for the soil. Chemical fungicides harm the plant, therefore alternative methods like natural fungicides can be used. Material used. Two fungus were used, Aspergillus niger and Fusarium oxysporum. Plant used was Xanthium stomarium. Two types of culture were used, liquid culture medium and solid culture medium, which were Richard's broth and Richard's agar respectively. Chemicals and other requirements were also used. The entire experiment must be carried out in aseptic condition with sterilized glasswares. Method. Firstly, the extract of different concentrations were prepared from 5 to 40%. Then the plant extract was tested in both the mediums along with the inoculated fungus. Biomass was estimated by drying the filtrate in oven at 180 degrees Celsius for 2 hours. On solid culture medium, the antifungal activity on solid medium was studied using agar well diffusion method. A control plate was maintained and the plates were inoculated for 7 days. Observation. In liquid culture medium, for Aspergillus, excellent growth was observed at 5% and the growth was reduced at 40%. For Fusarium oxysporum, growth was inhibited at 40%. On solid culture medium, Fusarium at 40% concentration showed the largest zone of inhibition. Results. In liquid culture medium, Aspergillus showed a considerable reduction in mycelial growth on 40% ethanolic extract, whereas Fusarium showed inhibition at 40% of the extract on the solid culture medium. For Fusarium, 5% shows overgrowth and 10% onwards there is a major inhibition seen. 40% shows maximum inhibition. Conclusion. The above investigation has brought us to the conclusion that ethanolic extract of Xanthium stomarium are effective against both pathogenic fungi Aspergillus niger and Fusarium oxysporum. Now I would like to conclude my presentation and thanks to our professor, Mr. Udebhan Yadav, for giving us this opportunity, guiding throughout. Thank you.
To present his paper, Identification of Microflora of Bacteria on Rusting of Iron.
Warm wishes to everyone. I, Aditya Sabe from Microbiology ID, will be presenting my research paper on topic Identification of Microflora on Rusting of Iron. This research paper was done under the guidance of Kunal Thakur sir. Now let's begin. Introduction. The maximum commonplace kind of iron corrosion takes place when it miles uncovered to oxygen in the presence of water, which creates a crimson iron oxide, generally referred to as rust. Rust can also impact iron alloys consisting of steel. The rusting of iron can also occur when the ions react with fluoride in an oxygen disadvantaged surroundings. Even as inexperienced rust, which is any other kind of corrosion, can be formed directly from steel, iron, or iron hydroxide. Rusting is the phenomena of the deposition of a reddish-brown coating at the surface of iron by means of the action of moist air, and the reddish-brown coating is known as rust. In simple phrases, when an iron atom is left in damp air for an extensive time, it gets covered with a pink-brown flaky substance referred to as rust. This phenomena is called rusting. What is the process of rusting? Rusting of iron is an oxidation reaction. During rusting, iron combines with oxygen in the air in the presence of water to form Fe2O3 H2O, hydrated iron oxide. This hydrated iron oxide is referred to as rust. As a result, rust is largely hydrated iron oxide, Fe2O3 H2O. The color of rust is reddish brown. Now let's see materials and method that were used during this research. There were two plates that were used. First one was sterile nutrient agar plate, and second one was sterile McConkey's agar plate. Nutrient agar plate with the composition of 0.5% of peptone, 0.3% of beef extract, yeast extract, 1.5% of agar, 0.5% of sodium chloride, and distilled water. McConkey's agar plate contains the essential nutrient required for the microorganism growth. Collection of the sample. Bacteria that causes rust was taken as subject sample for this study. Rusted samples were collected from different vehicles. One of the sample was collected from the 10 years old rusted bike parked outside the garage, whereas the another sample was collected from four-wheeler vehicle parked outside the police station. All the samples were collected in sterile containers and stored in sterile saline until they were delivered in the laboratory. The pathogens were maintained and the culture suspension was made and then propagated on the plates under septic conditions between the burners. Isolation of bacteria. Bacterial samples were spread on to the all three plates mentioned, nutrient agar plate, McConkey's agar plate, and PDA plate. The plates were kept for incubation at 37 degrees Celsius for 72 hours and further process was done. This is the result that we observed after 72 hours. Gram staining. Bacterial sample that was incubated at 37 degrees Celsius for 72 hours was taken out and the grown culture on media were used for staining method. Each plate culture medium was taken and gram staining was done by using different stains that are crystal violet, iodine, distilled water, decolorizing, safranin, and blood oil emulsion. After staining, slide was observed under microscope at 100x. This is the result that we observe under microscope at 100x. First one is under uh, nutrient agar plate and second one is of McConkey's agar plate. At last, result and conclusion. Different bacteria were isolated on different agar plates. Conclusion. These bacteria can be used demineralization and breakdown for future use. Thank you.
Now I request Mr. Udayban Yadav to present his paper. Topic is photosynthesis of silver and gold nanoparticle in peel and bark of plant pomegranate, Punica granicum.
Good afternoon, everyone. This is Udayban Yadav, Coordinator, Department of Microbiology, Interdisciplinary. Today's my topic is photosynthesis of silver and gold nanoparticles in peel and bark of plant Punica granatum, that is pomegranate. Photosynthesis of nanoparticles is an emerging area in plant science research. Different plants are used for this purpose, being it is the most eco-friendly and of synthesizing nanoscale particles of different salts. The plants have their potent sources of many valuable bioactive constituents, and these constituents contribute in reduction of salt in the system. A nanometer is 1 millionth of meter. Norio Taniguchi coined the term nanotechnology in 1974. Nobel laureate Dr. Horst Stormer said that the nanoscale is more interesting than the atomic scale because the nanoscale is the first point where we can assemble something, and it's not until we start putting atoms together that we can make anything useful. In present work of Punica granatum plant was taken as an experimental system for photosynthesis of silver nanoparticles and gold nanoparticles. Bottom-up and top-down approach are considered as the two most important modes of nanoparticle synthesis. Nanocages, nanofibers, nanotube, and nanorods are the specific types of nanostructures. Nanoparticles gives to the concept of biomedical nanotechnology, bionanotechnology, and nanomedicine. Several proteins make nanomaterials suitable for biotagging or labeling. Shape of nanoparticles varies from spherical to circular, oval, cylindrical, etc. Punica granatum plant is rich source in secondary metabolites, especially polyphenols such as alkaloids, flavonoids, tannins, and also steroids, triterpenes, etc. With lots of medicinal importance, Hence, this plant was selected. Proteins and enzymes present in plant extract are responsible for the reduction of metal ions in pomegranate. Thing about the materials and methods. Here, the plant material was taken as a bark and peel of Punica granatum. The chemicals: silver nitrates, chloronic acid, deionized water. The first step of preparation is a preparation of peel and bark extract of Punica granatum. 15 gram of each peel and bark powder were mixed with 300 ml distilled water, which allowed it for boiling for 20 minutes. After boiling it, filter it with the Wattman filter paper number 1. Use extract for future study. Preparation of 1 millimolar aqueous silver nitrate and chloronic acid, that is gold particles. 777.441 mg of silver iron powders and 177.223 mg of gold iron powder were accurately weighed and dissolved separately in 450 ml deionized distilled water. Next step is synthesis of silver and gold nanoparticle in pomegranate peel and bark extract. 25 ml peel and bark extract measured separately, and into this add 75 ml of silver and gold iron solution separately in a jar. Jar were agitated for few minutes and then incubate at room temperature. The method we have used for the analysis is UV vis spectra analysis, where bioreduction of silver and gold takes place as the time period of reaction mixers where increases. UV vis spectra analysis was done at the nanometer range of 190 to 1100. The next method is TEM analysis, that is transmission electron microscopy. Before time analysis, samples were sonicated for 15 minutes. A drop of sample was put on carbon-coated copper grids of 3 mm diameter. Then these copper grids were kept in infrared light for drying it. Then time analysis is done under electron microscope. About the observation and result, the data obtained on analysis of characters of phytosynthesized nanoparticles, nanoscale particles is tabulated in this topic. Table 1, which consists of for the average particle size, shape, and distribution frequency of phytosynthesized silver nanoparticles in Punica granatum, bark, and peel extract. Here the points are to be taken care of: the NTA analysis, where the mean size of silver nitrate peel extract is 59 nanometer and the mean size of silver nitrate bark extract is 61 nanometer. Then when during the NTA analysis, the particles per frame is 26.99 particles in silver nitrate peel extract and 37.66 particles per frame silver nitrate bark extract. In case of TEM analysis, shape of the particle is oval to spherical, and in silver nitrate bark extract, it is circular in nature. Under UV spectra, the study of silver nitrate peel extract is 463 is the maximum, and in silver nitrate bark extract is 400 nanometer is the maximum. About the table number 2, where the observation table for average particle size, shape, and distribution frequency of phytosynthesized gold nanoparticle in Punica granatum bark and peel extract. Here, the gold chloride peel extract contains mean size of 54 during NTA analysis, and gold chloride bark extract contains 58 nanometer during the NTA analysis, where the particle size per frame for gold chloride is 25.51, whereas gold chloride bark extract is 10.91 respectively. Then TEM analysis for the gold chloride peel extract is the shape of the particle is spherical, whereas the gold chloride bark extract shape is circular in nature. UV spectra under nanometer, gold chloride peel extract is 548 nanometer shows the maximum, and the gold chloride bark extract shows the at a 520 nanometer. The result and discussion about the table 1 observation table for average particle size, shape and distribution frequency and photosynthesis in pomegranate peel and bark. The same slide we have studied under the observation table. Here's the graph of the UVC spectra recorded as a function of time for the solutions prepared using a silver nitrate peel and bark of Punica granatum, where we can see the maximum spectrum. The figure 2 of UV-Vis spectra as a function of time required for the solution prepared using gold chloride peel and bark extract of Punica granatum. This image is of NTA analysis, where we can find out the particles per frame and average size of the particles. This image is of NTA analysis of 25% bark extract of silver nanoparticles. This photoplate 1ABC is of 10 image of silver nanoparticles showing spherical and oval shape nanoparticles in peel extract. This photoplate 2 is of 10 images of showing circular shape nanoparticle in bark extract. This NTA analysis of particles, where we can find out the particles per frame and the mean size of the nanoparticles. And this is the 25% analysis of bark extract of gold nanoparticle. This photoplate 3 ABC is a 10 image of silver nanoparticles showing spherical shape nanoparticles in peel extract. Here's the time to discuss about the result. The present work was conducted in the bark and peel extract of Punica granatum nanoparticles by treating with 1 millimolar concentration of silver nitrate and gold chloride. Nanoparticles in pomegranate peel and bark was done with the help of UV vis spectrometry, nanoparticle striking analysis, and transmission electron microscope. The result obtained in the present concluded as follows. In both the experimental sample, peel and bark extract of Punica granatum, it is concluded that the biomass used yields good response for the photosynthesis of silver and gold nanoscale particles. The UV spectrometry analysis revealed the spectrum status for both the samples in accordance with nature and solved use. Nanotracking analysis of silver nanoparticle indicates high frequency of nano-sized particles in bark extract compared to peel extract of the experimental plant. The average size of silver nanoparticles was more, that is 61 nanometer for a bark extract when compared to peel extract sample. The gold nanoparticle appeared with high frequency in peel extract. However, the size of nanoparticle was more, that is 58 nanometer in the bark extract sample compared to peel extract. The TEM analysis indicates circular, spherical, and oval shape phyto nanoparticles. The gold nanoparticles were circular in shape in bark extract, whereas in peel extract, sample of the shape of nanoparticles were spherical in nature. So this is all about the experiments. Thank you.
Now I request Mrs. Sonali Joshi to present her paper, Antimicrobial Activity of Different Hand Washes.
Hello everyone. I am Sonali Joshi, Assistant Professor of Thakur Sham Narayan Degree College. My today's topic of presentation is antimicrobial activity of liquid hand wash. As we know, hands perform many functions in our daily life and exposed to a variety of substances like dust, different bodily fluids, contaminated material, raw substances, and various stuffs from the environment during a personal hygiene. Most of the hand microbes can be killed using the hand sanitizers, but soiled or tinted hands with muck or organic material must be always be washed with the liquid hand wash. Studies suggest that using plain soap increases the spread of pathogenic bacteria to food by 3 times than using a liquid antimicrobial hand wash. Comparing hand washes with liquid with alcohol based solutions and washing with the antimicrobial for a median time of 30 seconds, each one showed that alcohol based hand washes reduces bacterial contamination 26% more than the antibacterial. So the materials and the methods are required for this practical is we take a liquid sample, that is a liquid hand wash. We prepare the cereal dilutions up to 10 raised to minus 4 of it. E. coli and the Klebsiella species, that is 1 gram positive and gram negative species, we use for the bacterial samples. And then the Muller-Hinton agar plate, that is a medium, is used for this. Miscellaneous are test tubes, sterile Petri plates, sterile cotton swab, sterile antimicrobial disc, and sterile pipettes. Then the procedure is, firstly, take the 2 test tubes and add 10 ml of liquid hand wash, that is 1 Dettol and other in one and 10 ml of Santur hand wash in other. By using water as a diluent, a series of dilutions is made ranging from the 10 raised to minus 1 to 10 raised to minus 3 of each liquid hand wash. Sterile Muller-Hinton agar plates were swapped with strains E. coli and the Klebsiella respectively. After that, aseptically deep different sterile discs in different test tubes containing the different dilutions of Dettol hand wash respectively, and transfer the disc in different quadrants of the Petri plate containing E. coli and the Klebsiella species. Do the same procedure for the Santur hand wash. The plates are transferred to an incubator and place it in incubator for 37 degrees Celsius for 24 hours. After the 24 hours, we observe the result like the undiluted sample of Dettol showed zone of inhibition of 12 millimeter in E. coli and 10 millimeter in Klebsiella, whereas Santur showed 16 millimeter zone of inhibition and 10 millimeter zone on E. coli and Klebsiella respectively. 10 raised to minus 1, minus 2, minus 3 dilutions of Dettol got 10 millimeter, 4 millimeter and 2 millimeter zones in E. coli, whereas it got 9 millimeter, 4 millimeter and 3 millimeter zones in Klebsiella respectively. It's the same was for the Santur. These are the results for the Santur and Dettol hand wash on the E. coli and the Klebsiella species. So from this, it is clear that the Santur hand wash is more effective on gram positive as well as gram negative bacteria than the Dettol hand wash. Thank you so much.
Now I request Mrs. Niyati Pandey to present her paper, Reuse and Recycling of Agar.
Good morning, everyone. I'm going to present my topic on reuse and recycling of agar, which is also done with my student, Ms. Ritishka Sharma, second year student of microbiology IB. The contents of the complete PPT goes as introduction, methodology, observation, results, and conclusion. We all know that agar, which was not even known earlier, was not used for the growth of the organism. In 1860, Pasteur became the first person to successfully grow bacteria in a lab setting. His medium was composed of sugar, yeast ash, and ammonium salts. He made significant contributions to the study of fermentation also. In 1887, a simple tool called petroplate came into existence and it had revolutionized microbiology. Agar is a component of cell walls in several species of red algae. Later on, this is a chemical polymer with galactose subunits that was used and that was dissolved in boiling water to form a gelatinous and solidifying agent when it is cooled down. This agar is basically the basis of our nutrition medium that we use to grow the organisms. According to our objective in this project and this measurement, the average Escherichia coli cell weighs about 2 picograms. And the loop-pool of the organism or the loop-pool suspension consists of 2 picograms of the cell weight. And the typical culture medium that we have on the plate consists of around 1 gram of nutrients in it, which means that all the nutrients that are present onto the petri plate are not utilized by the bacteria during the complete incubation period of 24 to 48 hours, which means that once the agar which is being plated onto the medium or is present into the medium is utilized by the organism, we discard it. So in that case, that discarded nutrient goes waste because the complete nutrient present in the medium is not utilized by the organism. So that is why in our current study, in our current project, we have studied whether we can reuse this medium and reutilize for growing the same species of organisms in it. Agar is traditionally used to harden culture medium at a final concentration of 1% to 2%. When studying motility, 0.5 weight by volume, anaerobic growth, 0.1%, and microaerophile growth, 0.05 to 0.5%, smaller amounts, 0.05 to 5%, and applied all the way into the medium. The agar, different types of nutrient agar that we used in our project were nutrient agar, McConkey's agar, Sabrod's agar. These were the three basic medium that we used in our project. We all know that nutrient agar, which is used for the cultivation of non-fastidious microorganisms as well as for quality control. So any type of microorganisms can be first grown onto a general medium, that is the nutrient agar. So we selected that medium. McConkey's agar is a selective medium as well as a differential medium, which is going to differentiate between the lactose fermenters, non-fermenters, as well as it is going to select only the gram-negative organisms to grow onto it. Then we selected Sabrod's agar, which is the medium that we will be specifically using for the fungal growth. So, which is used for isolation of both non-pathogenic as well as pathogenic fungal and yeast organisms. The methodology of the complete study and the complete project goes as shown here. On day one, we have prepared the medium, the agar medium. All the three types of medium were prepared separately. They were autoclaved and then they were put onto a petri plate. After solidification, the fresh culture, which is there into the in our laboratory, the laboratory fresh culture, which means the 24 hours old actively growing culture, was streaked onto this plate using a nichrome wire loop. And as we are talking, the nichrome wire loop is going to contain around 1 picogram of the cell weight of that organism. Now, this particular plate, which is streaked with the organism, is incubated at for 24 hours of 37 degrees Celsius at normal incubation temperature. Now, on the next day, proper growth is seen onto the plate. Now, this growth is scraped off using a glass slide. That is on day 2, using a glass slide. The colonies present onto the plate, that is the growth seen on the plate, is removed. And the conical flowers are filled with the jelly of agar, which is cut. Okay, so the colonies which are removed, the agar is then cut into small pieces and it is put into the conical flask. This medium, which is consisting of the reused medium or the used medium, is going to be autoclaved for the normal autoclaving time and temperature. Now, after autoclaving, the complete medium is poured onto a petri plate again, a sterile petri plate. And after solidification, the culture is streaked onto it, the fresh culture, the culture that we used on day 1, the same culture is streaked onto the plate and it is incubated again for 24 hours at 37 degrees Celsius. The same process is done for all the 3 different mediums in our study. The results that we see are that nutrient agar had normal growth of Escherichia coli on the first cycle of growth, that is the day 2 results. So we can say that day 2 media plate of Escherichia coli showed proper growth. On day 3, the growth was normally seen as earlier on day 1. So day 3 media plate also showed proper growth of Escherichia coli. Then Sabros's plate was seen growing normal Saccharomyces cerevisiae on day 1 growth of cycle as well as day 2 growth of cycle. So day 2 basically means the first cycle of growth, and the reused medium on day 3 also showed normal Saccharomyces growth onto it. McConkey's agar plate also showed normal growth of Escherichia coli onto it on the first cycle of incubation as well as the second cycle of incubation showed the same growth of organism onto it. The results of the complete study goes as follows. Microorganisms from the reused agar showed growth similar to fresh medium. A strategy of recycling culture medium is producing positive results. It demonstrates that the reuse of the medium is possible. The crucial factor that assists in the media solidification is agar. In this experiment, agar was easily recycled and no changes were seen until the second day. However, after 3 days, the color of the medium darkened due to the constant autoclaving, that is heating. Without any additional nutrients or without adding any additional nutrients onto it, the plates from the second and the third days demonstrated healthy development of the cells. So this means that to grow bacteria naturally, several metabolic products that inhibit growth must be purified. Okay, due to the trace component that is consumed during the first growth, all recycled plates have different colony sizes. So we conclude that reusing of the media is possible. It has had a big impact since metabolic activity by products from the microorganisms do not interfere with their ability to thrive onto it. The day 2 result demonstrates that the nutrition in the culture media was not fully utilized in the first cycle, and therefore we have reused the medium, and even after 24 hours of the incubation, it is still possible to grow the organism onto the same medium. Essential trace elements needs to be analyzed because they are going to definitely play a crucial role in the development of the microorganism. Agar, which is a major and expensive component of culture media, is also very easily recyclable in this study. So recycling medium can reduce cost, labor time, and environmental risks as well, which has brought to us on by bacteria genetic alteration because of that. Control of hospital infections, superpathogenic organisms can also be is also possible because of this particular reuse of the medium. So in case when we are not interested in just throwing the organism, or we are just interested in showing the students about the growth of the organism or the isolated colonies, in those cases we can just scrape off the growth of the organism and put the medium for reuse so that our nutrients are not wasted and our nutrients are reused for the growth of the similar organisms on the next stage. So future prospects of this study goes as after running 2 to 3 more cycles, we can check for more reusable media for more cycles. So we have just gone for 2 to 3 days of reuses or 2 to 3 cycles of reuse. We can check for more cycles of reuse there. Because of time limit, we had to stop somewhere. Other expensive media can also be checked for their reusability by this method. Like for example, we have expensive media like Harrison agar. There are other selective mediums like EMC agar, endo agar, blood agar, and all that. So this study can also be used, and future prospects of this study can go ahead by testing the reusability of those mediums there. So I end up with my references over here. Thank you, everyone.
Now I request Ms. Aishwarya Ajay Kumara to present her paper, Effect of Different Sterilants Used in Plant Tissue Culture During Micropropagation of Carrot and Black Mustard.
A very warm greetings to all of you present here. I, Aishwarya Ajay Kumar, will be presenting my topic on effects of different sterilants used in plant tissue culture during micropropagation of carrot and black mustard. Introduction. Tissue culture technique has been found valuable in the development of crops that provide better yield and more nutritious food. They tolerate drought, resist pests, and disease-causing organisms, and survive in harsh environments. Its successful application is in the improvement of various crops. However, this technique is faced with the problem of infection. Contamination is one universal problem in tissue culture studies, and it is experienced in all tissue culture laboratories and workers. Generally, there are 4 possible sources of contaminants. The plant, which may be internal or external. The nutrient media, which may be insufficiently sterilized. The air, and the research worker who might be inaccurate. While some contaminants are visible, which include bacteria, yeast, fungi, others are hidden or cryptic or are often difficult to detect, causing the most serious problem. So the maintenance of the aseptic condition has been identified as essential and the most important part of having a successful plant tissue culture. Now, the chemicals used in sterilization materials should be of effective kind, cheap, easily available, and non-toxic in nature, or else they can cause some kind of one that is strong enough to inhibit the growth of disease-causing microbes, even in small quantities, and at the same time do not injure the explants. Hence, it is important to determine the appropriate concentration of the sterilant and exposure time in order to sterilize the sequence, to standardize the sequence of using sterilants to minimize explant injury for better result. The materials and methods. First, the media. Full strength of Murashige and spoke media culture was used for this experiment with supplements, external supplements added to it in the form of glutamine, myo-inositol, citric acid and ascorbic acid, and sucrose and agar. The pH was adjusted to 5.8 using potassium hydroxide after addition of all these compounds. The only factor that was studied in this research limited to the explant material. So, the chemicals used. First is the sterilant 1, which was ethanol. Then second sterilant used is sodium hypochlorite. Sterilant 3 is mercury chloride, HgCl2. And the sterilant 4 is neem water, which contains oleic acid, stearic acid, and arachidic acid, and palmitic acid. You can see the procedures done for each steroid. Methodology. Fresh carrot or callus of a carrot was collected and kept at room temperature until the initiation of the experiment. The sample was peeled and cut to a size of 1 centimeter square. The sample was kept in a beaker or a small petri plate and washed with tap water. It was washed in teapot solution, decanted, and then washed again with sterile distilled water 2 to 3 times. Then it was washed with 70% ethanol for 30 seconds. Then washed with sterile distilled water 2 to 3 times. Then again washed with distilled water uh, 3 times. Then transfer the explant or the callus onto the sterile petri plate containing the tissue or the test tube, PTC culture. Then we sterilize the forceps using alcohol and then place the explant on MS media and we are incubated for at least minimum 3 weeks. The same method was used for all the other sterilants and the same procedure was done for black mustard seeds. You can see the observation of sterilant 1 showing slight discoloration of the media, indicating that there is a slight growth of bacteria happening over here. Then in sterilant 2, we can see the plant growth also quite visible. And sterilant 4, you can see a very good growth of the plant from the mustard seed. And we can also note that it has a good growth as well as there is less contamination seen in the media. Now you can see the callus of carrot, where you can see sterilant 1, 2, 3, and 4 visible clearly, while sterilant 2 has slight turbidity and sterilant 1 showing the maximum turbidity, indicating any kind of bacteria present during the culturing of this callus. Result and conclusion. The result of various effects of sterilizing agent on the contamination and color change or the browning of the tissue culture was noted. The percentage contamination was considerably higher in the samples sterilized with sterilant 1 and 2, which is ethanol, and now we can see the result and conclusions. Now we can see the result and conclusion. The result of various effects of sterilizing agents on the contamination and color change, browning of the mustard seeds and callus was noted. Percentage contamination was considerably higher in the samples sterilized with sterilant 1 and 2, that is the ethanol and sodium hypochlorite, than those that received sterilant 3 and 4, that is mercury chloride and neem extract, involving the use of mercurochloric and antioxidant solution of neem. When the samples were sterilized using sterilant 4, it proved better than those of sterilant 1, 2, and 3. In contrast to the contamination, the percentage of necrosis developed by the carrot was the highest. The sterilized mustard seed showed the least color change, with only one of them turning necrotic, with the highest percentage of survived culture. Based on the result of this experiment, the samples were consistently sterilized with subsequent studies using the different sterilants comparative, and the best kind of sterilant was observed to be neem water, which contained oleic acid, stearic acid, arachidic acid, and palmitic acid, as it did not even cause any necrosis and showed very good quality of growth in callus and seed culture.
Now I request Aditya Doble to present his paper, which is isolation and screening of human pathogens from different samples.
Warm greetings to one and all present here. This is Aditya Dhoble, and today I'm going to present my research paper, that is isolation and screening of human pathogens from different samples. I have done this paper in association with my classmate Aditi Tiwari and under the guidance of our professor Dr. Bharti Walekar, and my college name is delegates Thakur Shamnaran Degree College. First, starting with the introduction. Antibiotics are powerful medication that treat certain infection and can save lives when used properly. They either stop bacteria from reproducing or destroy them. Before bacteria can multiply and cause symptoms, the immune system can typically kill them. White blood cells, that is WBCs, attacks harmful bacteria even if symptoms occur. The immune system can usually cope and fend off infection. So the antibiotics we are going to deal with are gentamicin, vancomycin, streptomycin, erythromycin, chloramphenicol, methicillin, ampicillin. Starting with the methodology, isolation of pathogen. Urine pus and stool samples were collected from patients in Ghati, Aurangabad. 1 ml of each sample were enriched in nutrient broth and incubated at 37 degrees Celsius for around 24 hours, and spread bread technique was used. Pathogens were differentiated by lactose and non-lactose fermenting bacteria on McConkey agar. Further identification by gram staining and using selective and differential media identified by VTEC2 instrument. Antibiotic sensitivity test. The bacterium or a desired sample is swabbed onto the agar and the antibiotic discs are placed on top. The antibiotic diffuses from disc into the agar in decreasing amount, the further it is away from the disc. If the organism is killed or inhibited by the concentration of the antibiotic, there will be no growth in the immediate area around the disc. This is called the zone of inhibition. The zone sizes are looked upon a standardized chart to give a result of sensitive, resistant, and intermediate. Many charts have a corresponding column that also gives us the MIC, that is a minimal inhibitory concentration for that drug. The MIC is currently the standard test run for antibiotic sensitivity testing because it produces more pertinent information on the minimal dosage. Interpretation. Place the metric ruler across the zone of inhibition at the widest diameter and measure from one edge of the zone to the other edge. Use millimeter measurement. The disk diameter will actually be part of that number. If there is no zone at all, report it as 0, even though the disk itself is around 77 millimeters. Zone diameter is reported in millimeters. Look upon the chart and result reported as sensitive, resistant, or intermediate. Record the result for your table as well as other table into the table. So here are the pictures of the result. And in the third image, we can see the activity of various antibiotics upon the samples they put like pus, stool, or even other sample we are taking into account. In this table, we can see the various sources of sample of patient, that is urine, stool, and pus, and the activity of antibiotic against that particular bacteria present into that sample. And here we can see the antibiotics accounted are gentamicin, erythromycin, vancomycin, streptomycin, chloramphenicol, methicillin, and ampicillin. And here S stands for the sensitivity. That means that a particular microorganism is sensitive to that particular antibiotic, and R stands for resistance. That means that that particular microorganism is resistant to that particular antibiotic. So, identification of gram-negative bacteria based upon the various tests which were carried out, like ALA-PHE pro, aryl amidase, adonitol, L-abitol, D-celibiose, beta-galactosidase, H2S production, beta-N-acetyl glucosamidase, and D-glucose, and several other confirmatory tests were to carried out. And through this, three major gram-negative bacteria were isolated, which were Klebsiella pneumoniae, Escherichia coli, Pseudomonas aeruginosa. Similarly, identification of gram-positive bacteria based upon the various tests which were carried out like phospholipase C, xylose, beta-galactosidase, alpha-glucosidase, phosphatase, and cyclodextrin. Several other confirmatory tests were to carried out, and through this, two major gram-positive bacteria were isolated, which were Enterococcus califluorescens. So reference. For our paper, we took reference of many other paper, out of which 30% belong to China, 25% belong to India, 35% to America, and 10% to Africa. Acknowledgement. We, Aditya Dhoble and Aditi Tiwari, would like to express our gratitude towards our professor, Dr. Bharti Varikar ma'am, and our college, TSDC, for giving us an opportunity to present our research paper, thereby guiding and helping us throughout the process. Thank you.